the cell's function, the organelles, its function, its interaction, and how the replication is going to happen. Okay, so all these things can't be covered in one class. I mean, can be covered, but it will be too toxic for you guys. Okay, so I'm going to split it into as many as small sessions possible. So make the sessions more interactive. If you have any doubts, stop me. So if you have any confusions, or where you want some clarity, please ask then and there. Okay, it's up to you. Either you can ask at the end of the session, or you can stop me wherever you want. So cell, we number start pannom appadina, like most of you would have got an introduction of cell. Yeah. So from your perspective, what is cell? It's going to be interactive. Don't take it in other ways. Not a question answer session, not a Viva session. Okay. So I just want to know like what you know, so that I can fill in the gaps. Okay. So yeah. So what is cell? Anybody? What do you understand if somebody says cell? Something like that. Okay.
Cells are we are talking about eukaryotic cells.
Yeah, we are discussing about eukaryotic cells only. Okay, because most of our assays, other than infectious, is going to deal with eukaryotic cells. Okay, so we'll discuss more about eukaryotic cells. So, or be it even in prokaryotic, what is a cell? The function is going to be same. Its structure is different. Okay, its composition is different, but function is same. So the definition is also same.
Structural and functional unit of an organism.
Yes, it is the smallest. Okay. So long start. Which you have to remember. It is the smallest. It is a functional and it is a living. Okay. So the smallest functional living aspect of any cell, of any tissue, will be cell. Okay. So cell and everything now, like again you will have two things. One is prokaryotic, next is eukaryotic. Okay. So what is the difference between prokaryotic cell and a eukaryotic cell? Otherwise, what are the common components of a cell? In general, when somebody says cell, what do you understand? What are the things that is? What are the main two components that is present in a cell? Nucleus and cytoplasm. So you have one nucleus, okay, and you have one cytoplasm around it. Okay, fine. So with this, just to start, basically, like how does eukaryotic and prokaryotic cell vary in this structure? You have a nucleus. Inside nucleus, do you have anything? Come. Nucleolus. Okay. So inside nucleus, you have nucleolus. Okay. So does nucleus have a cytoplasm like thing? Nuclear membrane is this thing. So what you are seeing here, the part or the organelle. Organelle is again the smallest functional organ is called as organelle. Okay. Okay. The smallest functional aspect of or smallest functional organ is called as organelle. So inside the nucleus you have nucleolus. Okay. So nucleolus will be one organelle inside the nucleus, and there is a membrane. So what are these spaces made up of? What are these spaces made up of? There should be something, right? It can't just float. So this is called as nucleoplasm. Okay. Nucleoplasm. Just like how it is called cytoplasm, it is called nucleoplasm. So the nucleolus and the DNA material. Okay. Or we are talking about DNA cell here. So it is a DNA material or the nucleic acid material, to be on a broader perspective. So what is the nucleic acid material which we are talking about? What is the name in this? What is the name in a cell? How it is called? Chromosomes. Okay. Before chromosomes, how it is? Gene. Okay. Still more primitive form of chromosomes and genes. See, what you're talking about is one Okay, this is what? This is chromosome. Okay, this is chromosome, and you have lot of. Once you do a Gibbs banding or G banding, you will see all these bright and dark bands. These are nothing but the genes here. So each and everything will be one gene. So gene will be a small part of your chromosome. Fine. This much it is clear. Now, what is the primitive portion to this? From chromosome, you have gone to gene level. So what is the one level before chromosome? Sorry? Genome, otherwise called as still more. Exactly. Chromatids. Okay. So how is chromatids formed? Chromatids will be like noodles. It is unorganized, thin, slender nucleic acid material. Okay. When a cell is about to replicate, it will get condensed to form chromosomes. So chromosomes will again have a sister chromatids. You can divide equally the center. You have sister chromatids. Okay. And each arm of chromatids has its own, and this is all diploid cells. Okay. So you have genes everywhere. So this noodle-like thin, slender DNA or nucleic acid material is fully present here. Clear? So you have two things inside the nucleus. One is chromatids per se, which is the nucleic material, and you have nucleosome. Okay. So now, like, when and how does this get condensed into this? You have to get condensed, right? So first, for so much of material to be, placed inside a small area, you need to coil this or keep it in a organized way. Correct? So how it is being made inside a cell? Histones, per se. So histones are octamers. They are octamers, meaning there are 8 portions. This you are seeing from the front. So you have one more in the back also. So you have, it's an octamer if you see. So basically it is just like you have, just imagine you have 4 like this and 4 behind this. So there is a cleft in between. You can imagine, right? So in that cleft the chromatids are coiled. Okay. So that's how the long nucleic acid material is condensed into the nucleus. Okay. So in between you have this nucleosome. Nucleosomes are, if you see in properly dividing cells, you will see it as bright spots. Bright spots. So these are called as, in other words, they are called as NOR. Nucleus organizing regions. Okay, they are called NOR regions. So what is the significance of this NOR region? Now we are fully dealing about nucleus. Okay, we have not gone into cytoplasm first. So we have seen nuclear membrane, we have seen chromatids, and we can see chromosomes that is arising from the chromatids, and the chromatids or the chromosome arms will contain the genetic material or genes. Okay, so this chromosome, this long chromosome is coiled around a histone. Okay, histone protein, which is an octamer. Okay, why am I emphasizing octamer again? Because there are 8 parts, and each part has its own function in DNA regulation. It does not implicate your output. It does not affect your phenotype. Phenotype is how I look. Genotype is what I am made up of. Two different things. Genotype and phenotype. So if there is any change in histone, nothing of my outlook will be changed. Okay, but what I am made up of will be changed. So it will regulate the gene. So the histones are responsible for gene regulations. Each and every part of histone. For example, you have H1, then H2, then you have A and B in this, then you have H3, then you have H4, H7. All these things are histone molecules. Till H8 you have, because it's an octamer. Each and every part has its application in 2-3 major areas. What are the major areas that I am talking about when coming to replication? When somebody says one DNA is going to replicate or a cell is going to replicate, so what are the main areas or concerns which or main aspects which I am discussing? What I need, that is what you do in your PCR when you do a master mix.
Exons.
More than exons? Okay, since you have said exon, we will go into that thing first. So, if you see a DNA material in a linear aspect, this is a coiled aspect, right? This is coil. Now I am looking it in a straight linear. Okay, so it will have lot of areas, and there might be some areas okay. So in general, what all the things that I can see is, one is an exon, one is an intron, one is called a spacer. Okay, this is called spacer. Okay, so what do you understand by exon? What is the significance of exon?
Conserved region.
It's conserved region, or it is the coding region. Okay, so exons are nothing but coding areas. Then what are introns?
Non-coding.
They are non-coding areas. So what all comes under introns? Now exons, all the genes will come here, correct? Whatever any change in exon, there will be change in the gene. Okay, it will, because the coding area, you will code genes only. So when you code genes, your proteins or the genes are expressions changed. Okay, but introns are non-coding area. So any change in non-coding area is not going to affect your gene. So what is the significance of it otherwise? In other words, what it is made up of, or what all the components present in a non-coding area. Lesser form of protein, a more premature form of protein, still more. Somewhere in between. What or from where does a protein come? What is translation?
Transcription.
What is translation? RNA to protein is called translation. Where does it happen? It happens in cytosol. Sure? Pakka? No. Ribosomes. Okay, we'll come to that later. Keep it as a question mark, so we'll cover it. Okay. So coming into non-coding areas, it contains all your RNAs. Okay, what kind of RNA? You have small RNA, you have long non-coding RNA, okay, you have miRNA, that is microRNA. The other major forms of RNA, like somebody prompted, it is mRNA, tRNA, and one more, rRNA. Okay. So you have these three. Now what does this M stand for? Messenger. And T? Transport. R? Ribosomal RNA. Okay. So we'll see that also when we discuss the doubt. Where does the translation happen? So we'll discuss that. Apart from this, these are more functional RNAs. Okay. These are mostly seen in the DNA material. These are not seen in DNA material per se. They are products. When your DNA material wants, these proteins are formed. Okay. Again, the source is most of your rRNAs. Where does it come from? Most of your mRNAs. Where does it come from? And tRNA. Where does it come from? Because once you know this, then you will know how it works. All your ribosomal RNAs, the main area of production is these dots. What are these? Nucleosomes. Okay, these are nucleosomes. These are products of nucleosomes, and these two are in cytoplasm. Okay, clear? Oh, where were we? We were in introns. Okay, so all these three things, smRNA, long non-coding RNA and microRNA, these three are the common non-coding areas you see in a gene area. Okay, so now we have mentioned that it is a non-coding area. It is not gonna go for any DNA material. So what it is gonna do? Function and why it is there, and why they have classified according. எது வச்சு classify பண்ணிருக்காங்க? Small RNA, long non-coding RNA, micro RNA. It is depending upon the base pairs. Okay, depending upon the base pairs, they have divided this into like micro, small and long non-coding RNAs. Okay, now coming into these three things we usually don't do it here, be it NGS or be it fish or be it any molecular study, we don't do it here. But these are the areas now that is being under main priority, because again they don't affect your phenotype, but they will affect your genotype. That means they are responsible for your gene regulation. இதில் ஏதாவது பிரச்சனை வந்ததுன்னா நார்மல் ஜீன் ஃபார்ம் ஆகிருக்கும், ஃபங்க்ஷனில் வந்து you will have an issue. Correct? So they are like moderators or controllers that you see in a cell, which will ensure that your coded area from exons will work properly. Understood? Any confusion till now? So far is okay? Okay. And spacers are nothing but they are empty areas. They'll all be removed when there is something called post-translational modification of gene soldier. That is one thing. Okay, that we'll cover in detail when we do translation. Okay, so appo idella these spacers and all will be removed. Now what are few things what you are bothered when you are doing a PCR? What do you add which will imply your maybe like what to say, enhance your process in PCR? Enna venum? If I have to if this gene has to replicate, what should it have? This is a gene, right? We have assumed that it is a gene. Now it has to undergo replication. It has exons and introns. Fine. Now if it has to replicate, what should it have? Catalytic. So what is that catalytic thing what we are discussing? Promoter venuma. In order to start something you need something called promoter. Okay. And in order to continue something or make it better, you need something called enhancer. Okay, so these will be also seen in the gene material. Okay, promoters and enhancers. So now coming like okay, so we have seen what is inside the nucleus, what are the components of it, and how the coiling happens around the histones. Okay, then when we talk about a gene, what are the aspects it has? It has an exon, it has an intron. Exons are coding areas, introns are non-coding areas, and you need a promoter and an enhancer in order to continue the replication. Okay, so okay, this is one aspect. So imagine this is the cytoplasm. Okay, you have a cytoplasm bilayer and you have a, this is a nucleus. Okay, wow. I've just taken one quadrant of a cell. Imagine like a pizza slice. Okay, so this is your cytoplasm and this is your nucleus. So now like we have said, you have nucleosomes and you have the chromatin material. Okay. Now we are just going little bit into the cytoplasm. Okay. So what exactly is lying outside the nucleus once you come out of nucleus?
Organelles.
Nucleus, again somebody said nuclear membrane, which is a bilayer. Okay. We have 2 layers of it. Yeah. So outside vanda udane there is so what are these? Yes, endoplasmic reticulum, ER. Okay. So what are the types of ER you know? Smooth and rough. Okay. Why it is smooth? Why it is rough? இது ஒண்ணுதானா இல்ல டிஃபரண்டா? Are they both same or different?
Ribosomes.
Yeah. So you have ribosomes. Okay. Ribosomes attached to endoplasmic reticulum that makes it look rough. Otherwise, structurally, both are same. They are nothing but folded membranes into cisterns. Cisterns அப்படினா வந்து have you played this game? Like there will be 3 balls in one thing. You have to make sure the 3 balls goes into one. So it is all space connected. It is all one unit. Okay, and it all leads to one place. So there is one point of entry, one point of exit, but it is all looking like a maze. Okay, so it is a cistern-like connections that is present here. Okay, so where does it lead? What is the next step? Because there is a continuity. The organelles are placed in such a way the functions goes undisturbed in a systematic way. Okay, even the cell is designed so perfectly. Okay, it is not just lying or floating around. Right? So what is the next thing that is present on the other side? You will have one more cistern-like organ which is lying here. Exactly. This is a Golgi apparatus. Okay. Then you have okay. You will have one mitochondria. Then you have okay. So connecting these things, there will be something how is the cytoplasm, by the way? What is the medium of the cytoplasm? How it is? Solid, liquid, gaseous, semi-solid? Semi-solid. Sure. You want to do so much of reactions, movement in a semi-solid way. How do you assess proteins in general? When you assess protein, what is the common method you use? Which we are not doing here, but we were doing it once. Electrophoresis பண்ணுவோமா to assess protein. How do you move it? Over a gel. Everything is designed so only because cytoplasm is a gel-like material. It will take cells, it will take signals, it will take receptors, it will take protein in a undisturbed way. Solid la pona மாட்டிக்கும், liquid la pona எங்க போகுதுனே தெரியாது. Fine. Based upon all these research only, they thought okay, gel will do. Then they started with agar gel, then they developed the gels. Okay, so it is a jelly-like medium. Okay, it helps in 2 ways. One, it helps in taking all your materials from point A to point B in an undisturbed way, or in a more controlled way, I would say. Correct? Jelly la vandu you can't do unnecessary motions. Correct? Imagine yourself in a jelly-like environment. You can't do unnecessary motions. Okay, somebody is towing you, you have to just follow that route. Clear? Second thing, it adds little rigidity to the structure. Correct? It gives a shape, it gives a rigidity to the structure. So that's why it is a gel-like material. But still, you can't just leave it. You need some support also. So what are those things that is supporting the cells or the cell structure? What are those that is connecting the cytoplasmic membrane to the nuclear membrane to the organelle? You need something, like you can't just float. You need some foundation or you need some supports or slings type. So இதெல்லாம் பண்றது தான் வந்து your something called as cytoskeleton. Cytoskeleton. Fine. So cytoskeleton la enna varum appadina all your actin fibers, okay, your microtubules, okay, then your adhesion molecules, CAMs everyone, cellular adhesion, okay, the adhesion molecules, okay, all your adhesion molecules. So actins are nothing but they are proper fibers. You can just imagine like somebody doing bungee jumping and they have a sling attached to them. Okay, so it makes sure that it holds. It is not rigid. அவன் குதிக்கும் போது அவனுக்கு எவ்வளோ வேணுமோ அந்த அளவுக்கு போகும் அது, but it won't cut off. Clear? So that it holds all the organelles between one another. Okay. Another thing is tubules. Tubules are very small. They are like tube-like structure. இதுல ரெண்டு விஷயமும் இருக்கு. Like one, it gives a support. Just imagine a bamboo stick. It gives a support and also it helps in transportation of small molecules, selective molecules. Okay, so these are the things that is present in the interspread area to hold the organelles. Okay, now we were seeing something called ribosomes. Okay, that is RNAs. Your rRNAs. Okay. So now ribosomes, if you see, there are two types of ribosomes that is seen. One is nuclear ribosomes. Another thing is cytoplasmic or free ribosomes. Okay, ribosomes can also be found floating in the cytoplasm. Another group is found in the external areas of your endoplasmic reticulum. Fine. இதுதான் இங்க வந்து உட்கார்ந்துட்டு இருக்கு. In the cytoplasmic plane of the endoplasmic reticulum, you have these ribosomes, which are called as cytoplasmic ribosomes. தலைக்கு மேல போலல, it's okay la. Fine. So from the nucleus we said there is the nuclear ribosomes that is coming from the NOR regions. So இங்க இருந்து வர இந்த ribosomes, they just go through this, they diffuse and enter the endoplasmic reticulum. Clear? This endoplasmic reticulum is found all 360 around the nucleus. Okay, so nothing that comes out can bypass endoplasmic reticulum. Okay, it's a 360 structure. So once it comes out, inga ella vandu you will have lot of receptors and modulators. You have something called modulators. Okay, this modulators is like too much to discuss for now, but just imagine they will make sure, because whatever you are sending it here, ribosomes, they will go here, and in endoplasmic reticulum there is protein synthesis happening. Correct? Okay, these ribosomes, you know everybody that there are two ribosomes. Correct? Translation எப்படி நடக்கும்? Translation படம் பார்த்திருக்கீங்களா? So you have one small gully, one big gully, and in between you will have that DNA material running. 40S and 60S ribosomes. Correct? The S is nothing but the size of it. Okay. So one is 40 and one is 60S. In case of another one, it will be 80S. Okay. So in case of this, this particle alone will be formed and sent here. Okay. This assembly and the protein synthesis will happen here. So all the way when the protein molecule is moving, it will just form, and when it comes here, it becomes one protein. The ribosomes, whatever you have sent, will be converted into one protein here. Okay. Now this protein will be அப்படியே வெளியில, that is something called exocytosis. வெளியில அப்படியே வந்து இந்த ஒரு membrane ஆலயே போயிட்டு next it will go into your Golgi. Okay. In Golgi what happens is, you have heard something called, once protein is formed, there is something called protein folding. கேள்விப்பட்டிருக்கீங்களா? Protein folding கேள்விப்பட்டிருக்கீங்க. There are many diseases that occurs when the protein is not folded properly. It is called as protein misfolding அப்படின்னுவாங்க. Any common disease you have heard or you have come across because of protein misfolding ஆல வரும் அப்படின்னு சொல்லிட்டு. Alzheimer's கேள்விப்பட்டிருக்கீங்களா? Everybody would have heard Alzheimer's. Parkinson's கேள்விப்பட்டிருக்கீங்களா? Okay. The main cause of Alzheimer's is this. As the people age, okay, there all this quality control mechanism fails. Okay. So in Golgi, the protein folding is not apt, or it is not happening as per the requirement. You have misfolded proteins. In the misfolded proteins form ஆகிறதால தான் you have Alzheimer's, which goes and deposits in your cortical areas, causing memory loss. Okay. So protein folding happens, protein modification happens here. என்ன modification? Protein என்ன modification இருக்கு? Protein form ஆயிடுச்சு, அது என்ன modify பண்ணும்? What are the different types of protein? Sorry? அவ்வளோலாம் போகாதீங்க. Depending upon your macromolecules. Okay, macromoleculesனா நான் என்ன சொல்றேன்? Fat, protein, carbohydrate, இதான் macromolecules. கரெக்டா? இதை வச்சு, sorry, these are micromolecules. Micromolecules வச்சு like எப்படி நீங்க சொல்றீங்க? Just combination. If there is one fat and protein, what is called as? Lipoprotein. Okay, one fat and one protein, it is called lipoprotein. Okay. அதே மாதிரி ஒரு protein and sugar, glycoprotein. Okay. So இந்த மாதிரி complex proteins நீங்க பண்ணிக்கிட்டே போலாம். All those takes place here. It will only give ribosomes from here. The ribosomes will be changed into protein, okay, and these proteins are exported to Golgi where the modification happens. There are two things. Correct fold and the fold another, it will go and combine with a fat or a carbohydrate depending upon your body's requirement, and depending upon the location of the cell, the composition of these organelles change. If you see liver cells, you have more of Golgi because liver is very active. It itself secretes lot of enzymes. So enzymes are nothing but proteins. Also it metabolizes lot of enzymes, which is again proteins. So in liver it is seen more. Next, pancreas. Again you have lot of enzymes, both your exocrine, especially your exocrines. Yeah. So there also you have lot of these things. No, இதெல்லாம் பண்றதுக்கு வந்து you need something. What do you need? You have given a medium, you have given a route. Okay, it's like creating a marathon. Now this is a start point, this is your ending point, or I would say outside this to the tissue is the ending point, correct? For the protein to reach. What is required to do all these things? Something basic that is required to do all these things? Energy. Okay, va? அதுதான் இங்க இருந்து வருது. Okay, so it is involved in something called as oxidative phosphorylation. Basically, nothing but it produces O2, it produces ATP. Okay, all your enzymes, most of your enzymes are kinases. All your kinases require ATP. It might require the adenosine part or it might require the guanosine part. It might be ATP or GTP. But you need a triphosphate. The triphosphate is formed from mitochondria. Okay. So you have oxygen, you have ATP. So this is called as normally we breathe. The same thing happens. Gas exchange in lungs, alveoli, all those things. That is our respiration. This happens inside the cell. So this is called cellular respiration. Okay. So it involves in cellular respiration. Okay. So now the continuity is achieved, at least for the cell and the structure and the function. Okay, ta sollingla. So what are the things that is present in nucleus? Nucleolus, then chromatids, then you have a nuclear membrane. Okay, these three areas. So chromatids, that will all go into replication, that we will cover in a different class. Okay. Nucleosome, we have discussed, it is forming your ribosomes, nuclear ribosomes. Okay. So it will go and modulate itself, mature itself, become a protein and go outside by endocytosis or exocytosis. Very important, exocytosis. Okay. And it goes into Golgi. Golgi is protein modification. Okay. Protein modification. That is modification. Two things. What are the main two things that is happening? First thing is quality check. That is folding. Proper folding. Second, whatever the requirement is. Folding if you want a if you want a glycoprotein, it forms a glycoprotein or it forms a lipoprotein, depending upon whatever it is required. So that is this. From here again it goes outside, and there is change in the structure also. Cis trans in a structure. Okay. Whenever protein enters here, it will be in cis form. Okay. Whenever it is coming out here, it will be in trans form. That is the modification. That is the modification. Okay. Cis will be changed into trans by proper folding and by proper combination with the requirement as per the body's requirement. Okay. So this is one part. Inner purpose we have seen in cytoplasmic area, there are cytoplasmic ribosomes that is sticking onto the surface of the endoplasmic reticulum. Correct? Angiyum the same protein synthesis happens. Inga all the other proteins that is required for cell maintenance, cell function will be formed. What are the proteins? You have this actin fibers, microtubules, all these are proteins. Adoda maintenance, adoda upkeeping, plus cell each and everything is a protein. So adoda upkeeping la anga varum. Clear? Okay. So we'll stop here. This is just a basic of introduction to cell, the organelles, and also its one more thing we missed. So we made so much of things. So whenever we enter kitchen, we make so much of things. What happens at the end of the day? Mess. It becomes a mess. Correct? So somebody has to be there to clean it up. So that is done by this lysosome. That is lysosome and proteasome. Okay, va? So again, all these wastes are put in this gel-like material. Fine. Again, the benefit of gel. If it drops it here, it will stay here, isn't it? Isn't it or not? Fine. If it is a fluidic medium, it will just go somewhere else. But for example, one protein has come out. Okay, that could have seen the waste on this, it will be here. Now what this can do is, it will just go engulf all these things inside. It will go phagocytous or do endocytosis. Okay, it will just create a bleb and it will enter and it will have one and it will digest inside itself, so that no debris is there lying anywhere around, and it is also not excreting anything. It will just break down the products. If the products are reusable, it will reuse. Otherwise, it will just degrade into its minute tiny speckle. Okay. The same thing the proteasomes does. It mainly attacks the protein waste. Okay. Proteasome. Again, this is like this itself is one big topic. Proteasome ubiquitin pathway in I don't know how many have heard of it. Proteasome ubiquitin pathway. It itself is one big pathway where if there is any problem in this pathway, then all your unwanted proteins are all not taken back. Okay, va? So body will recognize that unwanted protein as also wanted protein and நம்ம இண்டன் போடுற மாதிரி அதுக்கும் இண்டன் போட்டுரும். ஓகேவா? இண்டன் போட்டு abnormal protein produce ஆகும். It will give abnormal function. Understood? Okay. So that we'll deal when we see the further classes. Okay. So we just close it here, and any doubts you want, you can ask me.